Hello everyone, this is Vivid Botany. Again we are encountering here the question about PCR technique. In my earlier video also I have discussed about the PCR technique. I have told you that PCR यानी कि polymerase chain reaction is invented by Kary Mullis in 1985 and he has been received conferred with Nobel Prize in Chemistry in 1993, and PCR is used to detect HIV suspected patients, mutations in genes suspected of cancer patients, is used to detect genetic disorders. The three are there. Once again I am writing here that PCR is used in three ways. One is to detect HIV. We know human immunovirus, retrovirus, then used to detect that is mutations in cancerous patients, and they used to detect genetic disorders. Yes. So these three aspects of PCR technique are based on single phenomena, that is amplification of DNA. Amplification of DNA means cloning of DNA. That is, when one dsDNA is given to the thermocycler, the given DNA is multiplied in many number of times in short span of time. That is, amplification of DNA is the main root cause of the three detections. How does this amplification is done? That is the concept of PCR. It is, if you see the process of PCR, it is done in three steps. The first step is that, let me write here, PCR steps involved. The first one is that, you see, this is double-stranded DNA, that is sample DNA that is placed inside the thermocycler. It is a computerized device for processing polymerase chain reaction. Let's suppose this is fifth end, third end, fifth end, third end. The dsDNA is antiparallel. So the two DNA strands are having the hydrogen bonds in between complementary nitrogen bases. We know that very well. Now these two DNA strands need to be separated for synthesizing the complementary DNA strands. So for the separation of two DNA strands, we need to increase the heat. We need to provide temperature about increasing temperature, that's about 94 to 95 degrees Celsius. At this temperature, the two DNA strands get separated. That is, the hydrogen bonds present in between the two DNA strands are broken down, and the double-stranded DNA will become here single-stranded DNA. Surprisingly, you should know that DNA is not denatured here. I mean, the single strand DNA is not denatured. Only the hydrogen bonds are broken down and the double-stranded DNA will become single-stranded DNA. So we can understand that up to 95 degrees Celsius also DNA remains alive. So that often we say that DNA is more stable to act as genetic material rather than rest of them like proteins and the RNA. So this process of increasing temperature and breaking down the hydrogen bonds to make dsDNA to ssDNA is called as denaturation. It is called denaturation. So this is the first step of the PCR technique. Now let us see what do we do in second step. In second step, each separated DNA strand acts as template to form a new strand. So here two templates are formed for which new strands are need to be synthesized. This is done first by attaching here a DNA oligonucleotide. Oligonucleotide is a short stretch of DNA or RNA that will, what do you call, polymerize, that acts as the starter for polymerization of nucleotides for in 5 to 3 direction. So this is called as primer. It's also called as here primer. What is primer? Primer is a short stretch of, generally it is 20 nucleotides. I write here NTS, a short stretch of 20 nucleotides, ssDNA or ssRNA is called as primer, that is attached to third end of template. That is here, this is fifth end of primer, this is third end of primer, this is fifth end of primer, this is third end of primer. So the short stretch of primer attaching to third end of template strand is called as here annealing. So I write here primer is attached to third end of template is known as annealing. ANNEALING. Right. Now what happens after attaching this primer to third end of template? The third step is that extension. Extension, this is. Extension. How do we understand this extension? That is, whatever primer that we have attached here, that will extend polymerization of nucleotides in template dependent manner in 5 to 3 direction like this. Template dependent manner in 5 to 3 direction. Remember always the DNA replication, transcription, translation, all that take place only in 5 to 3 direction. It will be never from 3 to 5 direction. So here this process is called as, the third step is called as extension. So in this extension, what happens here, that a DNA polymerase is, which is thermostable. This is DNA polymerase is thermostable. Thermostable means it can sustain high temperature like 94 degrees Celsius also it can sustain. So this thermostable DNA polymerase called as TAQ polymerase, TAQ polymerase. From which organism it is extracted? It's extracted from a thermophilic bacteria that is called as Thermus aquaticus. Thermus aquaticus. This is a thermophilic bacteria, Thermus aquaticus. From this bacteria, talc polymerase is extracted. This talc polymerase is used as the DNA polymerase in the extension process, that is from 5 to 3 direction in template dependent manner. Right? And like this, ultimately here 2 DNAs are formed. This is extension process. So by this what happens, after this is one cycle, like that after 30 cycles, by the repetition of 30 cycles here, 1 billion copies are made. Really it is very surprising to know that we can produce 1 billion copies by repetition of the same process 30 cycles. So the first step is here denaturation, I wrote it here. Second step is here annealing, and third step is the extension. In denaturation, the 2 DNA strands are separated by raising temperature 94 to 95, and annealing temperature is lowered where the primer is attached at third end of template, and in extension DNA polymerase will extend the primer in template dependent manner by polymerizing the deoxyribonucleoside triphosphates in template dependent manner. So for one cycle, one DNA is made. For one cycle, from one DNA, 2 DNAs are made. So here always remember the number of primers is equal to the number of DNA strands, that is from 2 primers, 2 DNAs are made. So we can write here number of DNAs that are formed is equal to number of primers. We have seen that here from one DNA 2 DNAs are made. So for forming 2 DNAs, how many primers are required? 2 primers are required. So that is number of primers is equal to number of the new DNA molecules are made. So this is the PCR technique. One more thing I would like to tell you here is that what is the main difference between the primer and probe? Often we call the term probe. What is probe? Actually primer, probe both are single stranded. Both can be oligonucleotide. Both can be DNA and RNA. Then what is the difference in between primer and probe? Generally, the primer is meant for extension of polymerization of the nucleotides in template dependent manner, whereas the probe is used to detect the gene of interest in hybridization process from the mixture of sample copies. So probe is to detect, I write here, probe is to detect gene of interest. Probe is to detect gene of interest in colony hybridization technique. We will see in biotechnology principle that to selection of transformed cells, one heading is there, one subtopic. In that, the last method is colony hybridization, where we make use of the probes. So probes are single stranded DNA or RNA of small stretches that are radioactively labeled and that are complementary to a small part of the desired DNA. They are used to detect the gene of interest by hybridization procedure in the sample of DNA, desired DNA segments. That is probe. Probe, do remember. Then what is primer? A primer is also oligonucleotide, a single stranded, radioactively labeled with the fluorescence either with DNA or RNA, but primer is used for elongation of the DNA, elongation of ssDNA in 5 to 3 direction by using that DNA polymerase in template dependent manner. So do remember generally that what is the difference between primer and probe. Now let's come to the question here, the PCR technique. PCR technique has been asked twice. PCR technique knocked our door twice during the NEET 2021. Why? Because it is an application of biology. It is a modern technique of biology. It's an evolution of biology. It's a landmark invention of biology. Today we are able to detect the COVID-19 with PCR only. We can't imagine what happens if there is no PCR. Today we are able to find out the genomic sequence of the different variants of COVID-19 virus because of the PCR. So I say that it's an evolutionary, remarkable invention of 21st century. PCR invented by Kary and Mullis. Right. Now he gave here the different processes, the 3 steps involved here, and he is asking what is the sequence of these steps. So the first one is here denaturation, second one is annealing, third one is extension. So the first step itself, the first option itself is the right option here, the denaturation, annealing and extension. Remember, in denaturation temperature is raised, in annealing temperature is lowered, in extension it is addition of complementary nucleotides to the template by using DNA polymerase, that is Taq polymerase. And remember, why should we use Taq polymerase here? Because Taq polymerase can sustain high temperature. Then why should we have high temperature? Because at that high temperature only the 2 strands of DNA get separated. At that temperature, if you lower the temperature, the 2 strands of DNA cannot be separated. Remember one more thing, DNA will remain as it is except the breaking of hydrogen bonds even up to 95 degree Celsius. So the option is here, one option, application part of the biotechnology. A very good question and very simplified. I think you have understood very well, and it can be asked that in which process the probe is attached, in which process the 2 DNA strands are separated, in which step that complementary, that is polymerization of nucleotides is done, or in which process the Taq polymerase is used, that is extension, in which process, in which step the primary is attached, that is annealing, in which process the hydrogen bonds are broken down, that is denaturation. Like that, we should have a good comprehension about the PCR technique, a novel technique, and we expect different kinds of questions on same PCR technique in next year also. So I think you have a clear glance of PCR technique. So let's be ready for knocking the next NEET, that is 2022. Thank you.